Thank you for the introduction and hello everyone. I'm really pleased you could join us today because we recently published a paper showing some exciting and long-awaited results, and I was due to present these at IABR 2022. However, due to a family loss, I was unable to attend. Therefore, when Alstone asked if I'd give a talk today, I realised, apart from a few of the analytical aspects which I've previously presented on, we hadn't actually presented any of the super exciting breath data to the breath community, which is kind of crazy. So as such, I'd like to thank Billy and Alstone for allowing us to share our work on the use of breathomics in an acute care setting and the discovery of exciting breath metabolite networks in acute cardiorespiratory exacerbations with everyone today. To start, I'd like to acknowledge the EMBA Consortium, highlighting equal contribution and special thanks to Rebecca Cordell at Leicester, who was instrumental in delivering the work alongside Wada Ibrahim, Matt Richardson for his expertise in advanced statistical analysis, Errol Gelia for supporting the pediatric branch, and our colleagues formerly at Loughborough, including Paul Thomas and Delia Salmon. Finally, I'd like to thank Professor Paul Monks, Chris Breitling, and Neil Greening at the Leicester Biomedical Research Centre and University of Leicester, and Professor Salman Siddiqui, who's now at Imperial College London. We understand the concept of exhale breath analysis, and that metabolites measured in breath reflect how our metabolism responds to genetic, environmental, and phenotypic differences. And if we look at this tree map from a recent review published only in August by Moore et al., we see the majority of breath biomarker studies target different cancers, with lung cancer taking the lion's share, approximately 52%. Then second to lung cancer is asthma, followed by diabetes and then COPD. And perhaps not too surprisingly, we see COVID-19 already appearing in the top 10 study pathologies. Therefore, from this chart, we can conclude that it is respiratory diseases which have been the focus of breath biomarker discovery studies. Looking at non-cancerous pulmonary disease only, we see asthma and COPD make up the majority, accounting for approximately 18%, with other diseases perhaps more related to infections, such as tuberculosis, and then cystic fibrosis, accounting for only 4% combined. Since today I'm reporting data on exacerbations of acute breathlessness in common cardiorespiratory diseases such as asthma and COPD, alongside pneumonia and heart failure, this is where we'll focus our attention on previously reported markers. To date, a range of individual compounds have been reported as biomarkers, including a handful of alkanes such as hexane and decane, methyl branched and cyclic hydrocarbons such as 2-methyldecane and cyclohexane. We also have the aldehydes such as nonenal-decanal, aromatics such as toluene, terpenes and terpenoids like limonene, and VOCs containing heteroatoms such as sulfides. I highlight these because it's important to recognize that several metabolites or VOCs have already been individually reported as breath biomarkers of inflammatory cardiorespiratory disease. And so when conducting a new study, in order to elevate and push forward the field, we shouldn't focus only on what we've discovered from scratch, which I will do today. We should also attempt to comment on the consistencies or inconsistencies of the data with existing markers, and whether that data can be used for the validation of previously reported compounds. Excluding breath tests which have been approved based on permanent gases and isotopically labeled CO2, the same message from multiple systematic reviews is that the development of breath diagnostics based on VOCs and its leap into clinic has got stuck behind this wall of inconsistency. The message often repeated is the lack of standardization in sampling and analytical methodology, which is important, but also trial design and outcomes, and fundamentally, the reason why we haven't been able to validate breath-based biomarkers yet is down to the inconsistency in the panel of the few individual breath biomarkers reported. So today I'm going to discuss the main findings from the acute data in EMBA and the discovery of breath metabolite signatures associated with severe cardiorespiratory exacerbations. However, I also wanted to bring to your attention a second discovery we made, which was the identification of chemically related metabolite sets in breath. Now, this was presented as the sort of second part of the results in the paper, slightly more hidden, but perhaps it's the more significant because it provides primary evidence of breath metabolite networks which could help address the inconsistency in individually reported markers. I imagine many of you are already familiar with the concept of a network. Whilst the use of powerful software for visualizing complex interactions in networks has only really emerged in the last couple of decades, networks and graph theory dates back to Euler in the 1700s, and this idea of describing relationships between different measurements and properties can be seen in the scientific works going back to Da Vinci. When reporting breath biomarkers, we acknowledge there is individual biological variability, and there's these confounding influences from our diet, lifestyle, and environmental exposures. And we try to focus on differences in the metabolite levels between the experimental groups of interest. But we do acknowledge the possibility of a complex interplay within the biological system, and that many of these metabolites may be highly interrelated. Then not only are the absolute metabolite concentrations between groups important, but also the relationships between these metabolites may be important for detecting pathological differences. Network analysis has been widely used in other omic fields and in metabolomics to show relationships between metabolites, enzymes, and genes through annotated pathways. But even independent of annotated pathways, just the clustering and networks allow us to better visualize intermetabolite relationships. Despite the range of biomarkers reported, there is little to no hard evidence demonstrating whether breath metabolite networks exist. There was one notable review by Isset et al., reviewed and published last year in April. Unfortunately, our work was under peer review at the time. They looked at VOCs previously reported as biomarkers and then grouped them by functional group. In doing so, they found that the association between VOCs were more consistent between cancer pathologies when compared with other disease states. So by taking a step back and assessing the markers already reported and looking at how those markers might be similar, they significantly increased the discriminatory power of their model for differentiating cancer versus non-cancer. Now, this isn't specifically looking at how markers might interact, but grouping compounds together based on a shared property is moving towards this idea of a network. They went on to conclude that the use of previously reported single volatile markers was unlikely to be effective. And I think that's a shared and widely accepted view within the breath community today. But also now we should probably be moving away from the idea that the reduction of the breathome to just a few individual metabolites is also not realistic, and in many cases will be difficult to validate considering the complexity and variation between cohorts. Perhaps this approach is still realistic for the studies of looking at infection and microbial VOCs, where you might get unique chemical markers being produced. Therefore, the previous review and other new studies are now sort of advocating for this concept of a small signature, using multiple VOC markers in tandem to build up a breath print or signature, increasing diagnostic power and overcoming systematic variability in comorbidities. Perhaps in combination with non-VOC related measurements such as lung function tests, and beginning to consider the associations between VOCs such as functional group. But the results presented here and today, I think, go several steps further, revealing distinct diagnostic signatures which reflect the complexity of the breathome while maintaining clinical utility. And they really challenge this idea of what we should expect from breath-based biomarkers. With these two objectives in mind then, I'll outline the clinical study, talk about how we ensured the collection of high-fidelity clinical and analytical breath data, and talk about some of our analytical methodologies, and then linked with Objective 1, show the performance of the VOC model benchmarked against the clinical data and blood markers, and then linked with Objective 2, discuss the result of the chemical similarity enrichment analysis. EMBER is the East Midlands Molecular Pathology Node, funded by the MRC and EPSRC, hosted at the University of Leicester and partnering with the University's Hospital of Leicester NHS Trust and Loughborough University. And with the move of certain people, the exec board now includes the University of Plymouth and Imperial College London. Breathlessness due to cardiorespiratory diseases accounts for more than 1 in 8 of all emergency hospital admissions. And despite the same presenting symptom, the etiology of acute breathlessness is highly varied, with diverse disease trajectories and treatment options. Diagnostic evaluation of acute breathlessness is highly reliant on investigations such as blood-based biomarkers, such as C-reactive protein, BMP, and radiological procedures. These biomarkers have clinical utility primarily in patients with single pathologies, but they have poor discriminatory power in patients with multifactorial presentations of acute breathlessness, and they're particularly challenging to interpret in the context of pre-admission treatment exposure, such as antibiotics for pneumonia and admission CRP values. Additionally, delay in blood sample processing at the point of triage can result in inappropriate treatment decisions and consequently harmful effects to patients. Therefore, we want to assess the use of breathomics as a point-of-care diagnostic in acute care setting. The primary aim of the acute study was to evaluate whether Excel VOCs can differentiate acute breathlessness in cardiorespiratory patients at triage, and because it was at triage, this was also evaluated in the presence of clinical diagnostic uncertainty. Patients were admitted to hospital and breath sampled within 24 hours of admission. The patients were also asked to come back up to 6 months later, post-exacerbation, for a follow-up visit in the recovery phase, and provide another breath sample, which we could use for monitoring longitudinal differences. Excel breath collection was performed in 277 patients, like I say, within 24 hours of admission, and this was across 5 subgroups, which included acute heart failure, acute community-acquired pneumonia, adult exacerbations of asthma, and chronic obstructive pulmonary disease against age-matched adult controls. And this afforded one of the secondary aims, admittedly we weren't necessarily powered for, which was to evaluate VOCs as associated with subgroups. The reliability of data is often assessed on statistical analysis. However, arguably, experimental design, how you conduct the study, and the discipline to maintain standards over a long recruitment period is probably way more significant for assessing the validity of the result than any p-value. Furthermore, the analytical data is often scrutinized, but the models you train and test the VOC data on are only ever as good as the original classification. And since this was an acute study where patients were being sampled on the point of admission, we had to account for diagnostic uncertainty by the senior clinical decision maker. Therefore, a clinical adjudication panel reviewed all the clinical case notes. They determined the primary diagnosis and marked their level of clinical uncertainty on a 100mm visual analog scale, or VAS scale. And they did this blinded to the original senior clinical decision maker's diagnosis, differential diagnosis, and any prior historical medical records. And this helped us gauge a metric for that uncertainty. Inaccuracy of the assigned diagnosis in breath discovery studies can jeopardize the scientific rigor and lead to overinterpretation of study findings. And this chart shows the predominant changes between the diagnosis on admission and the final primary diagnosis. And you can see here with the largest number of changes between COPD and pneumonia, which is perhaps as expected. And that increased uncertainty in these two conditions is reflected in the VAS score distributions. With this data, we were therefore able to test the performance of the VOC model, even in the presence of this uncertainty. So on to the analytical methodology. We used a combination of bedside sampling and advanced discovery platform to visualize the exhaled breath metabolites. Breath sampling was performed using a receiver to target and enrich the alveolar volatiles. 1 litre of breath was collected onto 10x Carbograph 1 solvent tubes. The samples were then dry purged within 48 hours, stored, capped in the fridge, and analysed within 6 weeks. Room air and air supply samples were also collected to monitor background VOC variation. These were collected initially with every sample. We then did a little analysis and noticed that there was little variation between samples collected on the same morning or in the same afternoon. So then they were reduced to 1 for every morning or afternoon sampling session. This inversely resulted in one of the largest hospital indoor air quality data sets. And so we also published our VOC concentrations in the room air, which could be of interest. Breath samples were analysed by thermal desorption, coupled with comprehensive two-dimensional gas chromatography, using flow modulation, coupled with dual flame ionization detection and mass spectrometry. Anyone who knows me knows I'm a big advocate of multidimensional gas chromatography, having developed as a technique in several application areas for the past 10 years or so. It provides unparalleled chromatographic separation of complex mixtures, making it an optimal discovery platform. For those of you not familiar with this technique, you can sort of think of one-dimensional GCMS as this piece of chromatography paper where you put the dot of ink on the bottom. As you run the water, the mobile phase up the paper, it separates the ink into different colors. Well, GCGC is the equivalent of turning that paper 90 degrees and getting an orthogonal separation. And so on seeing the GCGC plot here above, you can see we've got the primary retention time on the x-axis and the secondary retention time on the y-axis. And these two dimensions with the intensity data means you therefore get this nice 3D representation. And importantly, if you look at this GCMS chromatogram, these three peaks here, with the additional second dimension of separation, we can see for these three peaks, you actually can see that they are coeluting, and we get this additional resolution of this complex, well, not so complex mixture in this case, but if that's just what it does to 16 peaks, you can imagine what it does to 500 to 1,000 different volatile compounds. So I just wanted to squeeze in a GCGC chromatogram to show the typical separation of breath volatiles. So again, on this x-axis, we have separation due to volatility, and then on the y-axis, we have this orthogonal separation due to polarity or functional group, if you will. And we found that clinicians particularly liked the 3D representation of the chromatogram such as this, because it aided their interpretation of what we were actually measuring. They liked this idea that you could effectively image breath and visualize phenotypic differences in the breath on the chromatogram as you would on a scan. The breath samples were analysed in discrete analytical batches. This was a long recruitment period, and we needed a way to make sure the analytical data was comparable. At the beginning of each discrete batch, we ran a retention index standard, which allowed us to monitor retention shifts in both dimensions. Ultimately, the data workflow that we came up with resulted in less than 2% variation post-alignment of the internals of the reference compounds that weren't used within the alignment, and less than 3.5% variation in an external indoor reference material that we use over the 2.5 years. And considering that the RI, the retention index standard, was run every 6 samples, this equates to over a minimal variation post-alignment of over 2,400 samples, which is pretty impressive. The breath samples, air supplies and room airs were all analysed randomly and mixed up together. And then, like I said, at the end, we also ran every couple of batches this sort of independent air quality mix, if you will, showing the sensitivity and the elution order and changes in retention of common VOCs. We were able to use the peak area of the internal standard to monitor analytical performance, as well as the peak area of the acetone and isoprene within the breath samples. And they were pretty useful markers of when the sample had been messed up or mixed up. And when the breath acetone and isoprene levels didn't match the expected distribution for a breath sample, we were able to then also go back and compare with the receiver metadata, assess the portion of breath that was being collected, and determine whether the pumps had worked or if there was an error in the collection. Now, batch effects are a common issue in omics data or any analytical measurement. The existence of batch effects makes it challenging to compare data collected and analysed at different time points. So we sought to investigate the potential confounding effects which could possibly contribute to batch-to-batch variation. These effects included dates of major instrument maintenance, labelled here at the top as batch ID. We also investigated the operator that collected the breath sample, differences between operators, differences between time collected, so the point at which the sample was collected throughout the day, time stored wet before dry purging, and time stored dry after dry purging. And as you can see, the only confounder that had any batch variation appeared was major instrument maintenance events. Now, those who use advanced chromatographic instrumentation know that this is not necessarily surprising, and importantly, we were able to remove this variation from the data using parametric empirical Bayesian adjustment. And we're then able to visualize the before and after batch correction using TS&E. Now, I don't know how many studies collect this sort of data, but we did so that we would be in a position to assess and correct for potential confounding effects, even if there weren't very many in the end. We also routinely monitored the integrity of sorbent tubes, weighing them before each condition cycle. Every tube, mask and box was conditioned to reduce VOC backgrounds before use. We developed and provided SOPs to all the recruitment staff alongside regular proficiency testing and training. And then we developed custom software for capturing all this information and bringing it into a centralized system so that we could use it in the chemometric analysis. So I know the information just given can sometimes seem sort of an ancillary result, but having attended this conference now for a good number of years, I know there's a majority of people out there that still have an interest in how the data is being collected and the importance of knowing the differences in methodologies used. So moving on then to the discovery of the signatures of severe acute exacerbations and benchmarking that VOC model against the clinical data. Pre-processing of the GC-GC data resulted in the integration of 805 chromatographic features across the 277 patient samples. The peak areas of the chemical features were normalized and adjusted for confounding batch effects, as just previously described. Whilst data reduction is a crucial step in most chemometric and metabolomic workflows, in order to overcome noise, there is a risk of throttling the data. When you reduce the number of features to the extent where it becomes detrimental to the discriminatory power and wealth of phenotypic information originally afforded by measuring the whole metabolome in the first place. Therefore, the analysis plan and regression models used herein were chosen to mitigate this potential throttling effect. In order to create a concatenated list of exhaled breath biomarkers suitable for diagnostic application, we applied a threshold of 80% feature presence per patient group, below which features were removed. This left us about 730 VOCs. We then extracted the VOCs associated with acute disease using a relatively rigorous lasso regression, followed by a softer elastic net regression, and then removal of approximately 38 peaks that were considered to be chemical and material artifacts, such as siloxanes, based on their prevalence in the procedural blanks. This generated a final panel of 101 exhaled breath volatiles. Following the regression analyses, linear combinations of predictive features were used to produce breath biomarker risk scores. Therefore, we significantly reduced down the number of VOCs to identify a set of markers associated with acute exacerbation. And within the 101 VOCs, we found smaller panels, approximately 15 to 25 VOCs, which were associated with each of the 5 subgroups. Furthermore, all the chemometric analysis was performed blinded to the clinical diagnosis. And one of the questions we'll always ask is, what do we actually do with all that room air data and air supply data that we generated? Because everyone collects it, but it's not always clear what everyone does with it. So we performed exactly the same analysis and generated the same model. So we were asking the same question of the room air data and the air supply data. Therefore, any VOCs discriminating the patient groups in their room air, we know were also arising from differences in the environment and not inflammatory response. We were pleasantly happy to see that only 2 VOCs were found to have stable non-zero regression coefficients in the room air and air supply, one in the air supply, which overlapped with the pneumonia score, and one in the room air, which overlapped with the healthy control group. And this really highlights the robustness of the selective feature separation models that we chose and the care taken in the sample collection. Of the 101 breath VOCs, therefore, 60 were identified to MSI level 1 standards against pure reference standards with matching primary and secondary retention times and mass spectra. A further 20 were identified to MSI level 2 against matching mass spec and RI library management. So that's almost 80% of a panel of 101 VOCs identified with a very high degree of confidence. And then because of the group type separation afforded by GCGC, the remaining 22 unidentified VOCs could still be assigned a chemical class based on their MS and 2D retention times. Alongside that, we then ran a 7-point calibration curve in triplicate for actually 84 different compounds, which included the 60 identified by standards, therefore providing at least semi-quantitative data for the significant majority of the VOCs. To achieve an unbiased discovery of VOCs predictive of acute breathlessness, patients were block randomized post hoc into discovery and replication cohorts. Randomization allowed internal replication of the diagnostic breath biomarkers while adjusting for relevant confounders. The value of the generated VOC risk score was found to be significantly higher in acute cardiorespiratory patients compared to healthy volunteers. For the replication cohort, the same VOC risk score differentiated participants with acute disease from the control group with an area under the curve of 0.89, a sensitivity of 0.79, and specificity of 0.85. Using the clinical adjudication results, each patient was assigned a degree of clinical diagnostic uncertainty with that visual analog score, the VAS score, at the point of triage. Diagnostic uncertainty was defined herein as patients with values higher than or equal to the upper quartile of 20 millimeters on the VAS score, represented here by the blue bars. The acute disease biomarker risk score was able to identify acute disease with high specificity and sensitivity, even in the presence of diagnostic uncertainty. Using linear combinations of the most stable features from the multinomial regression model, a set of scores was produced for predicting the probability of belonging to the different disease subgroups. As not to bore you with countless ROC curves, here's just a summary table generally showing high specificity but lower sensitivity. Again, this lower sensitivity is not surprising actually, and kind of encouraging given that we're looking here at acute versus acute disease. And the dysregulation observed in the breathome is most likely from biological responses which are shared across acute exacerbations such as systemic inflammation. Now, I really like these plots. The green lines show the balanced accuracy of the biomarker risk score for acute breathlessness, which was 96% in the discovery and 75% in the replication. And so to check if this data is not just all voodoo, we asked what would happen if you were to randomly shuffle the patient labels. The reason we did this is because clearly the ROC curves for the discovery cohort at least suggest we're seeing some discriminatory effects simply because of the number of variables and sample size. In other words, we remain skeptical because of the potential to overfit the data. Therefore, this is a really harsh but quite nice way to test the structure observed in your data. And you can see the accuracy for the randomly shuffled data is the red line. And you can clearly see the true class labels are significantly higher than the randomly shuffled data. So that was a bit of a relief, and I'd encourage you all to do, or at least try this at least once with some of your data. So topological data analysis, or TDA, is an unsupervised machine learning tool, and it's been used for the analysis of large-scale, high-dimensional data. It's highly sensitive to patterns that are often overlooked by other data reduction tools. And TDA, in this instance, allowed us to visualize how VOC risk scores were benchmarked against the clinical blood-based data. So in this bottom left-hand corner here, this is a TDA plot. Each circle or node in the TDA map represents a subject or collection of subjects. If two patients are very similar in their VOC data, they're put together on the same node. And if two nodes have enough in common, then they're connected by a line. And therefore, patients with similar breath VOC data grouped together. The plot of the discovery cohort shows 4 topologically distinct networks or groups of subjects. And when we overlay the patient classification, we see these correspond with different subgroups. Here we have colored the networks by the proportion of acute COPD exacerbations in each node. So non-COPD patients are scaled blue, and the patients labeled COPD are then scaled red. And you can see that pneumonia and COPD exacerbation subjects occupy the polar ends of this same node. So the VOCs in the discovery cohort show that there is some structure in the data with COPD and pneumonia lapping. And then we can color the same plot, but instead by the average values of CRP and BNP, which are two of the blood markers measured in the study. Subjects with normal CRP and BNP values are blue and scale upwards towards red with increasing values. And so you can see a high CRP in the COPD pneumonia cluster, and you can see high BNP values corresponding topologically with the heart failure node on the right there. If we then repeat the TDA in the replication cohort, we can see healthy volunteers and patients with acute heart failure form distinct topological groups. And the respiratory emissions due to acute asthma, COPD, and pneumonia form this topological continuum, but with distinct regions within the cluster. And again, if the networks are colored based on the average CRP values and BNP values, despite the merging of the groups, we can see high CRP values correspond with the COPD pneumonia cluster, and the high BNP values correspond to that topologically distinct cluster of the heart failure patients. The takeaway from these graphs is that the exhaled VOC data yield topologically distinct clusters that distinguish the underlying causes of breathlessness, and it corresponded with the blood-based biomarkers used in both the discovery and replication cohorts. And that brings us nicely to the final aim of the talk, moving on to the exciting metabolite set enrichment analysis and the discovery of breath metabolite networks. We wanted to find a way to go beyond simply reporting yet another panel of potential breath biomarkers which are potentially inconsistent with other studies. And so we asked ourselves the question, how can we generate further evidence from our data that these VOCs are indeed associated with acute exacerbation? For example, studies looking at oxidative stress cells have shown the production of low molecular weight branched hydrocarbons. Therefore, subsequent breath studies that have found branched hydrocarbons amongst their panel of markers often attribute them as products of oxidative stress. For example, in this study, we identified several branched hydrocarbons. But is that sufficient? Is the identifying hydrocarbons sufficient? It's not actually provided any evidence that these compounds within the data are coming from the same biological response. Yes, they're both hydrocarbons, but we can extract more from the data to tell us if they're coming from the same response or source. Unfortunately, detailed mechanistic understanding using pathway analysis is not applicable here because metabolomics databases are poorly annotated and volatilitic databases are even worse. But fortunately, there is a middle ground here between simply reporting markers of interest and that full annotated pathway analysis, and that is metabolite set and chemical similarity enrichment analysis. So what does this mean? So statistical independence among metabolites is incorrect. We know multiple VOCs can come from the same metabolic pathway, produced from the same enzymatic reaction, or even from the same confounding source, such as room air. So in the example here, if we identify highly correlated metabolite sets, then we overcome the multiple hypothesis testing problem. And instead of testing 6 hypotheses in this particular instance, if the metabolites can be treated as a single set, then we're only testing 1. As I said, pathway analysis isn't available, but instead of using biochemical pathway annotations for mapping metabolites to identify these metabolite sets, a more logical way herein is to use the molecule's chemical structure itself. Any markers in breath arising from the same pathway would be expected to be highly correlated, show set level significance, and be up or down regulated together. And they're also likely to have very high chemical similarity. Therefore, to move away from simply reporting this list of individual biomarkers, we performed this chemical similarity enrichment as a first step to mapping VOCs back to the disease response. Just to mention, this work was inspired by the work of Dinesh Barapul and Jeremy Ash on correlation network analysis. And since grouped variable selection was inbuilt in the models that we used for the feature selection in the data processing slide I showed earlier, our data workflow allowed for the identification of sets of possibly correlated features that are predictive of outcome. So what is this chemical similarity enrichment analysis? So starting with our 101 VOCs, so each circle here, each yellow circle represents an individual VOC. For each one, we generated raw p-values and fold change. We were then able to define sets of highly correlated metabolites using hierarchical cluster analysis. And then for each metabolite set, we used a KS test to determine the set level significance, providing p-value for the total set. So by comparing the distribution of the p-values of the individual VOCs within that correlated set, we could get a significance level for the metabolite set itself. Since we were able to identify the majority of VOCs in this study, we were also able to calculate a chemical similarity coefficient based on the these binary chemical fingerprints that are available for each chemical. And so instead of just being like, oh, we've got two hydrocarbons, they're chemically similar, we're actually able to put a quantifiable metric against chemical similarity and use the Tanimoto chemical similarity coefficient to compare the chemical similarity of different metabolite sets. So this was performed for every metabolite set. So for each set, we have a set level p-value, a fold change for the set, and also a metric for chemical similarity. You can then plot the set level significance of each set against the chemical similarity, with each circle or node representing a set of metabolites, the size of that node representing their fold change, and the color of the node representing the up or down regulated, and the appearance in the plot being either that they're statistically significant or those that don't appear in the plot aren't statistically significant. And so using this technique, we're able to identify significantly enriched, highly similar metabolite sets, which we can then link back to the core finding. When applied to the 101 VOCs identified herein, 20 metabolite sets were identified, 11 of which were significantly up or down regulated during acute cardiorespiratory exacerbations, as depicted by the circles here. Of the 7 metabolite sets that were upregulated, sets 3, 5, 7, and 9, were all predominantly acyclic and branched hydrocarbons, with set 7 given an example here. So, furthermore, we can look into the set, and for example, set 7 is clustered towards the right, based on its Tanimoto coefficient, which is that way of quantifying chemical similarity. And we can see 2-methylbutane, 3-methylpentane, a couple of dimethylpentanes, hexane and cyclohexane. So I think you'd all agree that they are very, the Tanimoto coefficient has done a good job at identifying that chemical similarity. I wanted a better way to show these metabolite sets and chemical information, so I came up with this. Now bear with me. Here we have a radial cluster diagram, if you will, and I was able to plot each of these metabolite sets in the circles or the nodes on the left onto this tree. So in the middle, we have all the different metabolite sets that were identified. Then the purple bars represent the log p-value between acute and control. We then have the fold change represented by the red and blue bars. And then on the outer ring, we then have the chemical names colored in the background by their broader chemical classes. So if we zoom in, we can see correlated together a bunch of C8 to 10 hydrocarbons, all elevated in acute breathlessness. Several of these have been reported individually as breath biomarkers, such as decane, 2-methylnonane, and dimethyloctane. However, in each study, in each independent study, a different one has been reported. And so we see that inconsistency of markers reported across the studies. By taking a step back here and looking at how these metabolites interact, we can see herein they are highly correlated and clustered based not only on functional group, but also carbon number and substructure. So this is providing the first evidence that they are likely arising from the same source or same biological response in acute exacerbation. Also on this slide, we can see the C1 to 2 sulfides, C3 to 5 carbonyls and oxygen-containing VOCs. And within that group, we can see solvents, IPA and DCM PET, and methacrylene and methylvinyl ketone, for example. We can then see we have the C5 to 7 hydrocarbons, all very similar structure. That's that set that I showed you earlier on, all clustered together and significantly elevated in acute breathlessness. Then we see all the terpenes clustered together, which aren't particularly discriminative, which makes sense. And then we have C4 thiols and sulfides all downregulated. This is interesting. Perhaps it's related to differences in diet between the controls and the acutely ill people. We then have a large cluster towards the left-hand side of C11 to 16 hydrocarbons, again, predominantly elevated in acute disease. So in summary, results of the metabolite set enrichment and chemical similarity analysis show there is significantly enriched co-expression of C5 to 7, 8 to 10, and 11 to 16 hydrocarbons, as well as C3 to 5 oxygen-containing VOCs with high chemical similarity. And these exhibit the highest change during acute exacerbation. So this provides primary evidence of exhaled VOCs indicative of disease response measured in vivo. Now, we wanted a way to sort of look at this data differently and think about how can we validate this. The structure observed in data suggests the presence of metabolite communities in breath. And we know biological networks can possess community structure properties, which is the basis of a network. So we turn to a community detection technique called Lavane clustering, which looked again to identify significantly enriched metabolite sets. When applied to the breath VOC data, we are able to produce a network graph based on the Lavane clusters identified. So each node in this graph represents a VOC, and each color of the nodes represents a community of correlated metabolites. We were then able to interrogate each Lavane cluster, and so you can see I've pulled out this green cluster here. We found that the metabolite sets generated using this completely independent method, looking at modularity between networks as opposed to chemical similarity, were concordant with the chemical similarity enrichment analysis, but only broader. They provided broader metabolite sets, as you can see here, capitulating both of those significantly enriched metabolite sets identified before. So this was helping to reveal the potential discovery of metabolite networks in breath. And interestingly, we've been able to validate these networks and metabolite sets further by assessing their longitudinal changes in the same patient cohort, and also look for them in pediatric exacerbations. And so that data will soon be made available. So we've met the two objectives of today's talk. I've covered in detail about how we went about discovering signatures of severe acute exacerbation. And then for the second objective, hopefully you've seen how we've tried to challenge current expectations of breath biomarkers. Now, it's not perfect and there's still lots of things that we're looking to build on, but I think we at least have attempted to push the field forward in how we think about breath data and possible ways for addressing the inconsistency in biomarkers reported. So in conclusion, we've established breathomics as an acute care platform for cardiorespiratory research. And we did this by developing robust clinical and analytical workflows. And this resulted in the identification of high diagnostic sensitivity and specificity of biomarkers of acute exacerbation. Alongside this biomarker identification, we are able to discover significantly enriched high chemical similarity metabolite sets co-expressed in acute exacerbation. And these results reveal new insights into the breathome and the potential identification of breath metabolite networks. Thank you very much for listening. If you have any questions, please don't hesitate in contact. And also, if you have any potential collaboration ideas, again, feel free to reach out. Thank you very much.
Excellent. Thank you so much for that. That was really fascinating and great to see the results of such an extensive study. So I commend you and the whole team for this fantastic work. We had a few questions just to kick it off. At the outset, you mentioned some of the challenges within the field of what has been previously reported, and it was really great to see the extent that you guys went to get confirmed IDs for some of those VOCs, so MSI level 1. I'm wondering if you could comment more generally on what you observed when you were kind of looking into the literature in terms of quality of what has been reported, separate from the idea of being able to group VOCs together into these networks.
Yeah, so I mean, by no means an authority for sort of commenting on standardization and things like that, but I guess I wouldn't necessarily comment on what the literature has, but what we felt was necessary to ensure that sort of analytical rigor and also clinical rigor in the collection of data. There's so many different variables that can affect your data, and quite often you're not in a position to control it, because quite often breath is sort of an opportunistic sampling. It's sort of bolted onto another clinical outcome that they're trying to find. So even if you can't control all the factors, at least trying to identify whether they've had an impact on your data quality, such as operator of a sampler, time stored, time of day that has been collected, just looking at those factors and seeing how they impact the data, like we did the batch correction, for example. So I don't know if that's a routinely utilized data processing method. I know a few groups use it and things like that, but we took the steps that we felt were necessary to sort of ensure that we knew what we were seeing in the data was indeed what we were hoping to see, not just artificial sort of separation, I guess.
Brilliant. Next question relates to the idea of using networks and grouping compounds together. So you mentioned some of the limitations in standard metabolomics, but obviously metabolomics and blood-based metabolites have been very well studied over 100 years, and there's pathways there. Have some of these techniques been developed and applied in standard metabolomics? I know you referenced one paper there, but maybe you could discuss in a bit more detail if these techniques have had success there, and therefore how they can be applied in breath.
Yeah, I mean, correlation network analysis has been applied in more traditional metabolomic fields, I guess blood-based biomarkers. Yeah, and that's where we received our inspiration from. We thought, what are these guys doing to sort of improve it? And unfortunately, a lot of the stuff in the literature is pathway analysis, where you need decent annotated pathways. That to be honest, actually for blood, even the annotations in blood are, I would say, are limited. They're definitely extensive compared to the breath, but even then, the shortfalls in the annotations there, because a lot of the technologies and data approaches adopted from metabolomics are taken from genetics, which again has even more years of research behind it. So we're at a disadvantage by not being as established, but we can sort of be inspired by the techniques that they're using. And so yes, correlation network analysis is, I wouldn't say it's standard, but I would say people are using it in metabolomics, and therefore we want to see what would happen if we applied it to breath. And this sort of data-driven, instead of relying on the pathways, using the chemical data that we actually have to hand instead, and seeing what that tells us, because we do have that data, because we went to the effort of identifying as many of the metabolites as possible, because even with the unidentified compounds, we still knew what chemical class they were, and we still had a mass spec for those, so we could still get some idea of chemical structure, whether it was like a branched alkane or something like that. So we could still input that into the analysis and get that information.
I like that idea that some or all of them can relate back to principles such as oxidative stress, where you had things that have been piecemeal reported in the past. But obviously, in the application of patients who are acutely breathless, you mentioned at the outset that the goal would be to have something that could rapidly triage patients in the clinic, essentially. I was wondering if you could comment on the translatability of what's been found to fieldable instruments or solutions that can be used in clinic.
Yeah, so obviously it makes the idea of a breath signature perhaps more complex, but I don't think that complexity voids the clinical utility at all, really. I mean, if you look, for example, at how polygenic risk scores are used for estimating sort of a cumulative contribution of loads of different factors, that's sort of the approach we're taking here. We're trying to use maybe a mixed model to simultaneously assess all the variants at once, and so I think the data science is there and able to sort of facilitate that. From a technology perspective, how that actually implements the translation of biomarkers to, say, sensor-based technologies. I think you're left with a few different options. If the power really is in sort of monitoring multiplex signatures, either you have more of a service-based setup with a more not discovery, but more of an advanced platform behind the scenes, or you develop a more advanced sensor array that's actually capable of handling these multiplex sensors. And I think as a discovery scientist, we often focus the difficulty being on the identification of the biomarkers in the first place. But I reckon the translation of that technique to a sensor, even though we've seen examples of that done for exposures and stuff, for a multiplex sensor that's very specific, I think that challenge is as big as identifying the markers in the first place. So I think it still has clinical utility, but I think it changes the way we have to think about that.
Yeah, and it all starts with biomarkers themselves. Once you have those, you can move on to step 2 of figuring out how to detect in field. One question here coming in from the audience, just as a reminder to people, please post the questions in the Q&A button at the bottom. So the question here is, I see value in evaluating correlated metabolites. However, to what extent do you think the grouping highly similar chemicals could cloud relevant signal, as the ratios could be more informative as a reflection of metabolic pathways than the combined abundance of similar molecules?
Yeah, so that's not to be confused. So what we're not doing here is assessing the combined abundance of molecules, because you lose that granularity. What we're doing is assessing those interrelationships, basically as a mixed metabolite signature, and assessing how, okay, some might go down, some might go up within that cluster. Generally, though, from the same source, they tend to move in the same direction, but still you might have offsets in those intermetabolite relationships. However, even as a crude sort of just assessing total group area, actually the only breath test, I think, to be validated based on actual VOCs and not an isotopically labeled gas or anything like that is Phillips's total alkane curve. So as crude as that sort of approach sounds, actually, I don't know if it's a coincidence then with the sort of discovery that of breath metabolite networks and things moving together that actually works. But no, that's if I understand the question correctly, we're not looking. We still maintain the granularity with the individually resolved markers and assess that. We're not doing total group sum of an area.
Yeah, you can see the individual fold change in p-value for those group VOCs on your plots.
Yeah.
One question here relates. I think this relates back to discovery type approaches. So let me just. Is it more promising to create a breath-based diagnostic device which is based upon breath metabolite networks, where the whole volatile, where machine learning and AI is applied to large data sets to distinguish healthy and unhealthy populations?
So my interpretation of this question, it kind of fits this question always comes up, and if we just use pattern recognition or sense technology, can we just throw more patient numbers at it and just distill the signal that way? And I'm an advocate of no. I think you, as much as you increase your signal, you also increase your noise. I think it's important to recognize what is actually happening and what metabolites are involved. So I think it will help to an extent, but I think you need, obviously more patient numbers is always going to be more useful. However, I think you do need to understand what is happening. I think it's a combination of the two. You have your discovery studies and you have your implementation of sensors, but I don't think you can just throw more patient numbers at sensors. I don't.
Yeah, I tend to agree. I think it's been attempted many, many times before. None of it is translated, and understanding what the molecules are, relating back to the biology, puts you in a strong footing to then, as you say, translate into point of care sensor-based solution if that's actually what is required. We have time for one last question. Actually, maybe two. So one, for some of the VOCs, could you comment on the concentration range, so the ppm or ppb levels, and to what extent it's important?
So breath has a very dynamic concentration range. So things like acetone will be sort of in the ppm level. I was just trying to look back to exactly where I sort of report those concentrations. But most, if you plot their concentration range against all the sort of metabolites, you sort of get poison distribution. Like you get one or two that are absolutely dominated, like acetone and isoprene in the ppm range, and then most things sort of then trail off into the ppb, and then if you have the detection limits, into the ppt range.
Yeah, brilliant. There's one question about standardization. I think we can hold off until the panel discussion tomorrow on that. Mike is joining the panel discussion tomorrow afternoon. And standardization is one of the topics of discussion there. So we have time literally for one very quick one. Do you have any plans to study acute exacerbations in fibrotic diseases such as IPF?
I don't right now. However, that's not to say I couldn't, or that shouldn't be done, but not right now, no.
We'll make sure to connect you to the person who asked the question there. Excellent. So thank you so much, Mike, for taking the time out to present the very exciting work. We really appreciate it, as I'm sure the whole audience do. So thanks again.
Cheers, buddy. Thank you.